Thank you. Thank you very much for the kind invitation. I would like to start by a big word of congratulations. It's a great meeting, and we have all partners in the room, and I would like to emphasize how nice it is to be in Dublin, and you do very well, and congratulations on that. I'm going to present, so I think the timer is not set at the right time. I was told I was speaking more than that, but I can speak 14 minutes if you wish. But I'm going to discuss basic science and translational medicine. So it's interesting because I think my big message is a message of hope. I'm going to show you that over a very long time period, when people understand the condition, when people understand the mechanisms of the condition, when many people working in different setting partner, you can end up with a very positive outcome. Okay, I have some disclosures with pharma, and I'm a medical doctor from Université Paris-Saclay, and I work both in the clinic in a rare disease program. I work also in the European Reference Network for rare lung diseases, and I have a laboratory entirely devoted to a condition called pulmonary hypertension. It's not a disease, it's a condition. It's characterized by ongoing elevation in pulmonary pressure, pulmonary artery pressure. So it's not the systemic hypertension, it's hypertension within the pulmonary circulation. So you cannot measure it with simple tools. You need either to estimate it with echocardiography or measure it very precisely with a right heart catheterization. And this condition named pulmonary hypertension has many variants, including rare variants, which are extremely interesting to analyze because these variants have discrete mechanisms with targetable pathways. So that will be the focus of my talk. I wanted to say that the stories are always very long, and we always have to look behind. And we have to recognize that many, many pioneers have worked in the field. I mean, all the fields we discussed today, sometimes you have names with disease. You have to remember that these doctors, etc., they describe things, and then other people built on that and then discovered the genes and then tried to find a solution. It's the same thing in pulmonary hypertension. In 1960, the WHO organized a meeting with some Nobel Prize winners who described the first measures of pulmonary pressure with catheterization. And at that time, there were some conditions, chronic lung conditions, which led to what we called cor pulmonale. So increased pulmonary pressure due to chronic respiratory disease. So at that time, they described many information regarding the normal pulmonary pressure, the abnormal pulmonary pressure, and the consequences. But there was no discussion whatsoever on the mechanisms, the cellular and biological mechanisms. Then in 1973, there was another WHO conference in Geneva because there was an outbreak of pulmonary hypertension due to appetite suppressant intake. This occurred in Switzerland, in Austria, and in Germany. And this was incredible because a very rare variant of pulmonary hypertension went crazy. And there were many cases in 3 countries where there was a diet pill marketed. And then people thought about the mechanisms. And among the molecules, there was a serotonin, there were other molecules, and it was very interesting. Then it took 25 years to have another World Symposium, and then every 5 years we meet with all the people, patients, pharma, doctors, basic scientists, to try to find new frontiers and new ideas in the field of pulmonary hypertension. And the first time we had a task force devoted to cellular and molecular pathobiology of the rare form of pulmonary hypertension called pulmonary arterial hypertension. It was in 2003 in Venice, and I was privileged to be the lead author on that. And we worked a lot at that time. We made a lot of PowerPoint figures. It was not beautiful like a bio-render picture, but we had a lot of ideas. You see here on the left, we were describing what could be the targets for new therapies in pulmonary hypertension. Then we described the serotonin story with this diet pill story. Then we described the gene causes of some familial causes of pulmonary hypertension, and then we discussed gene environment interferences. And on top of that, we understood better and better that patients with pulmonary arterial hypertension had a disease of abnormal pulmonary artery physiology. They had constriction and obstruction of the small pulmonary vessels, and this leads to pulmonary hypertension, and then it will lead to right heart failure, exercise limitation, disability, and death. So the very first mechanisms which were easier to identify were mechanisms of dilatation and constriction of the vessels. When you constrict, it's difficult to pump through the circulation. When it's dilated, it's easier. And there were many targets, and these targets were not little things. I mean, many people got Nobel Prize awards for the description of these mechanisms. For example, Sir John Vane described prostacyclin, which is now one of the major drugs for PAH in the community. And you had also all the people who worked on nitric oxide, and you know that drugs like sildenafil, tadalafil, and others, they can be used for patients with PAH because they will relieve the constriction and favor the dilatation. And on the right, you have another pathways described by Japanese colleagues, endothelin-1, which is the most potent vasoconstrictor. And thanks to the endothelin receptor antagonist, it was possible to relieve the pressure, to relieve the burden on the right heart. And the translation, and I thank you for inviting me to discuss translation because it went very fast. I mean, a Nobel Prize winner in 1982 translates into a New England Journal of Medicine paper in 1996 with then the approval at the same time. So I think for the many people in the room who suffer from rare diseases with the feeling to be orphan, I mean, we were orphan. There was no more orphans than us. Nobody cared about the condition we were dealing with. But thanks to scientists, thanks to community of patients, thanks also to the commitment of very few people, there were some deliverables, very visible deliverables. And you see many, many articles in the New England Journal of Medicine, and each time a drug approved. I mean, for us, it's a great relief, but it's not enough because we don't have a cure. So we are invited sometimes to summarize what is the current state of the art in pulmonary hypertension. And you see this was 20 years ago in the New England, 2014 in Circulation, always the same cartoon. A vessel, 3 pathways, and we can target 3 pathways. That was the story. We can target 3 pathways, and the vessel will dilate more, they will constrict less, and the patients will do better, walk better, feel better, survive better, not sure, but certainly when you combine drugs together. So that was important, but of course, when you look at our large registries, in France we have a PAH registry with more than 18,000 patients for conditions which affects around 50 people per million, so it's a very comprehensive registry. The median survival is around 5 years, 5 to 7 years. So there is a long way to go, especially when you know that the condition affects people in their 20s, 30s, 40s, 50s, so any age. So 5 years is not much. So we need to think outside the box, and we need to see what was not targeted among the pathways we described 20 years ago. And the TGF-beta pathway is important. So transforming growth factor beta, very complex, very redundant. I'm not going to make a very complex description, but this is meaningful in terms of remodeling. So the remodeling means the growth of the vessel. This superfamily is involved in development. It's no longer vasoconstriction, vasodilatation, it's more growth, and that's very important. At that time in 2004, it was not possible to consider targets which would be treatable in that pathway, and we needed a community of researchers meeting every 5 years, as I told you, simple people, normal people meeting with a common goal to discover what is wrong in PAH. And what is wrong in PAH is clearly a remodeling. It's not constriction. Constriction is important, but what is important is why are these vessels so growing inside their wall to obstruct progressively the lumen of the vessel. And you see here a normal pulmonary vessel is very thin, very, very thin, single layer, and it inflates over time when you exercise. So there is no constraint, no resistance to flow. here you have major resistance to flow. And you have to understand how can it happen. And it's very complicated, of course. But this is one of the many pathways which are dysfunctional in pH. And I will not describe that in great detail, but it's a question of balance, always a question of balance. In pulmonary arterial hypertension, you have an imbalance which favors remodeling, which favors the growth of the small pulmonary vessels. And in normal individual, you have what we call a pulmonary vascular homeostasis. And our idea is to understand what is triggering the remodeling in order to directly target what is wrong in these patients. And this question of balance can be summarized in simpler cartoon. And you see that there are two pathways, one blue, one red. The red one triggers proliferation. The blue one promotes anti-proliferation. And in pulmonary hypertension, you have a problem in some patients. You have some mutations here. And you have a mutation in the pathway which protects you against proliferation. So a mutation against something which protects you will drive proliferation. And you see here in pulmonary hypertension, in familial forms of pulmonary hypertension, you have these abnormal signaling through what we call BMPR2, and this will lead to proliferation. And clearly now we understand better and better what we could focus on and what could be targeted in pH. And clearly many people worked on that, and of course many people worked with different mindsets. And I love that cartoon. It's a bit complicated again, but this is the problem. The problem is the balance. The problem is the balance. So either you want to promote what is dysfunctional in pH, and you want to trigger anti-proliferation, and in that case you try to cover what is not functioning in pH. So you could try to stimulate the BMPR2 pathway with activators, with gene therapy, with recombinant protein. But there was another mindset, and the other mindset was it's a balance. And indeed, in pH, you have too much triggers on the TGF-beta activin nodal branch, which is the branch which stimulates growth. And here you can try, rather than stimulating, you try to oppose this pathway. And there are many ways to oppose the pathway which promotes growth. And among the many drugs which were in development, there was a drug which is a kind of ligand trap, which tries to block this pathway and try to restore the balance, not by pushing up here, but by blocking the other side. So it's very simple. It took 60 years to be developed, but now people have really identified one of the many pathways which could be targeted with a reverse remodeling action. And this is a biologic agent called sotatercept. And this agent is very simple. In fact, it is an immunoglobulin G1 fragment with the receptor of activin. And activin is one of the main drivers of proliferation. And there is excess activin in people with PH. And you see this little drug acts as a ligand trap, you see? It blocks activin, and it will try to restore the balance between anti-proliferation and proliferation. And that's the story. We started with that. It was 6 or 7 years ago. We didn't know COVID was coming, but we started a program. And we had some friends in Brigham and Women Hospital in Boston, Paul Yu and other. And on an animal model, they showed that this little drug, this ligand trap, was able to modify all the parameters which are disturbed in experimental models of pulmonary hypertension. But there is a long way from animals to humans. But at least here in Science Translational Medicine, there was this beautiful article showing that in many experimental models, it was possible to reverse remodeling. And this was, for us, another trigger to develop research and do clinical trials. I told you, John Vane, 1982, New England 1995. So here, in less than that, we started a program. And the program was very, very straightforward. We took patients who were on best standard of care. We don't want to make me too. We want to make a significant add-on effect on top of established therapies. And in PH, I didn't mention that in my introduction, but we currently have 14 drugs approved for PH, so 14, 1-4, which is a lot for a rare disease because there is a lot of research, and it's, I think, rather not easy, but it is rather logical how to interfere with the pathways. And here you see the patients were not many, only 106 patients, all of them treated with best standard of care with 1, 2, or 3 PH drugs. And you see that add-on, sotatercept, on top of these established therapies allowed dose-dependent reduction in the resistance to the blood flow in the pulmonary circulation, measured directly with the right heart catheterization. Then, on top of that, of course, we measured more direct benefits, because what we want patients to feel better, function better, survive better. That's the 3 very simple words. And here you see that on placebo, there was no change in PVR and dose-dependent reduction. And on top of that, the walk distance improved by 20 meters, and other parameters were improving. There were side effects. Among the side effects, the most important one were increased hemoglobin. And it's interesting because this is a repurposed drug. We discussed this morning repurposed drug. This drug was initially developed for anemia. And for anemia, you increase hemoglobin. So our patients increase their hemoglobin. So it is a side effect, but not unexpected. There were some unexpected side effects, including thrombocytopenia, and interestingly, telangiectasia. So it really means that we deal with a drug which disturbs angiogenesis. And our patients, in fact, you will see that later, they develop telangiectasia, they develop nasal bleeding, so they look more and more like a hereditary hemorrhagic telangiectasia patients. So we modify a phenotype, and that's really fascinating. Based on the phase 2 trial, you know, phase 2, you can call that proof of concept. Then you have phase 3, you call that pivotal. And the pivotal studies are designed to demonstrate a benefit which will allow approval and reimbursement and hopefully improvements for patients. So there were 3 major studies in the phase 3 program. The first one was called STELLAR. And STELLAR is very simple. You take patients on best standard of care, you treat them with sotatercept, it's a sub-Q injection every 3 weeks, or placebo, and you measure a very simple measure like 6-minute walk distance, and you see that the 6-minute walk distance did not improve by a small margin, but by 40 meters. And on top of that, there were secondary endpoints. You know, we always have secondary endpoints, and we rank them by order, and we analyze them in a hierarchical order. And you see that all these secondary endpoints were positive, except the very last one. And finally, these patients improved many, many parameters, including time to clinical worsening, which is a reflection of outcomes. The side effects were what I told you: nasal bleeding, telangiectasia, increased hemoglobin, and low platelet count. And last week, Monday last week, I was at the American College of Cardiology. I'm a respiratory doctor, but I go to cardiology meeting. And I presented for the first time the other phase 3 trial, which is, I would like to thank the patients very, very, very clearly, because it is a study on sotatercept in patients with pulmonary arterial hypertension at high risk for death. So it is a difficult study because you have patients who have more than 15% risk of dying at 1 year. I'm not going to go through the details. I don't think that's the setting where I should go to that. But I think what is important is that we were very careful with ethics in an outcome study like that with very hard endpoint. And we predefine an interim analysis after 59 events. And 59 events means 59 patients who died, who were transplanted, or were hospitalized for more than 24 hours. So we had patients enrolled, and once again, we thank them very much. The patients were on maximum therapy, you can see that most of them were on triple combination therapy, and despite that, they had a very high risk for death. They were, for most of them, on transplant program, and this is the endpoint. It's very rare to see an endpoint like that. This is time to major morbidity, mortality event. This is the active drug. This is placebo, and of course, after 59 events, the DMC. DMC means Data Monitoring Committee demanded the study to be terminated, and all patients rolled over to active drug. It's rare to have that in a rare disease, and I did not want to insist too much, but we had only 86 patients per group, and the results were read at less than 1 year. So clearly, the quality of the investigation is emphasized on that, and you see the reduction was 76% versus placebo. So I don't want to be too detailed, but the deaths were less common on drug versus placebo, transplantation less common on drug than placebo, and hospitalization less common than on placebo. You see here, that's what we call forest plot. All the subgroup analyses favored the active treatment, including patients with scleroderma who are very difficult to treat. And the secondary endpoint were also ranked in an order, but here, of course, because the drug was terminated early, the power was not strong enough to show statistical significance, but the editors of the New England told us it's not important, you terminated the study for good reasons, just show the data and the confidence interval, and I'm not going to spend too much time, but you see, even though death was not statistically significant, it was reduced by 58%, and death or lung transplantation reduced by 66%. We don't have to wait for statistical significance, and we can discuss that later. So adverse events were the same. There were no new adverse events in patients with advanced disease, always this HHT-like phenotype. And I just wanted to say that for me, it's a landmark trial because it's a milestone. It's the first time a PH study is designed to assess major morbidity mortality events, and it is the first time we stop a study early for efficacy. So that's important, and we even stopped the whole program. There were other phase 3. We stopped it because it was considered it was not ethical to expose people to placebo, and we will maybe in the future discuss that. The last thing I wanted to tell you is that you need to be lucky in research. So sotatercept, ligand trap, I don't want to make a long story, but a long story short, it's an activin receptor type 2A ligand trap. There were other companies, very strong companies, which developed activin receptor type 2B ligand trap because it was supposed to be safer. And this is very interesting because this drug, which was engineered to be safer, was tested in a phase 2 randomized control trial, exactly the same. It's a copycat of Pulsar, of the phase 2 of sotatercept. And during the winter, pericardial effusion occurred in patients. And the study had to be terminated because of pericardial effusion of unknown mechanisms occurred in these people. So in rare disease, even when you think you have understood, you have engineered the drug as well as you could, you can be surprised, and that's what happened. So my last slide will be this slide. I mean, I'm delighted to have a fourth pathway in PH because now we have a new area for research, and this fourth pathway is the TGF-beta pathway. And certainly there will be other drugs in the future, but right now there is only one, and it is interesting to see that it delivered quite well. So thank you to the patients, thank you to the scientists, basic scientists, translational scientists, healthcare providers, and also to the industry, which takes some risk to develop drugs. So thank you very much.